give it to her. Hi, I'm Heather. It's really, really great to be here. Hi, Betty. Abby is actually gonna present the project to you. I just wanted to introduce myself and tell you that it's a really great pleasure to be here. I really look forward to interacting with families. I love it, actually. I like to squeeze children and I like to hear your stories. So please don't hesitate to come talk to me, email me, call me, reach out to me if you have any questions that I can help with. I'm a veterinarian and a researcher, and my whole entire lab focuses on using large animal models to treat human genetic diseases, including this one. And so Abby today is going to talk to you guys about what we're doing to make a model so that we can study the disease, but then also get a therapy that's going to actually work and work through all the trials and tribulations that happen, because we're not rodents. And while the rodents are very useful, it's really helpful in my experience to have an intermediate model before you go to human clinical trials to optimize and make things as good as they can possibly be. And with that, I will give you Abby, Dr. McElroy.
Hi, I'm Dr. Abby McElroy. So I'm also a veterinarian in Heather's lab. So originally we were working on a pig model, but we've switched to sheep, which is really what we're best at as a lab. So that's the point of the title here. So as Heather said, large animal models are kind of the intermediary between mouse work and getting to human trials. And what's interesting about our lab is that our animals live completely normal lives as farm animals. So they live outside on a normal farm at Tufts. They're not housed as lab animals. So they experience normal herd dynamics. They experience heat stress, cold stress, insects, anything that a child would experience out and about in the world, they experience. And that's something that we're really committed to. And it's also just a way to better recapitulate the human experience, which is very different from mice who live a very sheltered life in a lab. So we as a lab are very, very good at generating large animal models. So these are some of our current models that we either have on the ground or are in development. So this cow, I don't know if I have a pointer, was given about $1 million worth of gene therapy to treat maple syrup urine disease, and she now is essentially normal. So she lives on our farm. She's very happy. We call her Val, short for Valine. And she is a very, very sweet girl that we all love very much. And then the Tay-Sachs sheep are kind of the flagstone project of our lab. And we're working on our ACTA2 model for you lovely folks. So initially we had planned to use pigs because there was some thought that there may be a vascular advantage to using them. There was some thought that they may be more similar to the human vasculature. But as you can see from these images, the sheep and the pigs are actually essentially identical in terms of their cerebral vasculature. So that's the pig, that's the sheep, very similar. So in terms of studying the cerebrovascular disease, there's not an advantage to using pigs. And as I said, as a lab, we're quite good at working with sheep. Another advantage to sheep is the difference in the genome between the sheep and the pig. So this is the nucleotide sequence around the 179 mutation for the human. The top is the pig, the middle is the sheep, and then down here is the amino acid sequences. So the pig has a greater number of mismatches to the human sequence than the sheep does. So the sheep doesn't have as many highlighted nucleotides, so it's more similar. So that makes it a little bit easier for us to generate the model. And then the protein sequences are essentially the same. So essentially, in the beginning, we were focused on base editing and prime editing. So I'm sure many of you have heard of CRISPR. That's where you just go in, you make a double strand break in the genome, and then the cellular machinery repairs it. With CRISPR, you have a bigger risk of off-target effects because you have to repair essentially a greater portion of DNA. With base editing and prime editing, it's a CRISPR-based method, but it's paired with an enzyme that makes a single break only in one strand of DNA, and only one single nucleotide is switched out with the nucleotide of interest. So you're just taking one single letter and replacing it with the letter that you want to replace it with. So it's a much more targeted approach, and you're much less likely to get off-target edits. So you're much less likely to create a problem somewhere else in the DNA. And this could be used for introducing the mutation or for fixing it at a later date. So the problem that we had with this method, and we do have some strategies for potentially fixing this later on if we go back to it, is that when we tried to implement it in cell culture, so using sheep cells grown in the lab, we were finding that we were getting quite a low efficiency of editing. So these bright green cells are edited cells. So our editing ratio was probably around 10% with the base editor and the prime editor. And then when we would isolate and sequence the DNA, we weren't seeing that any of the DNA was edited. So we were trying to introduce the 179 mutation. So this again, this was our base editor. You can see very few green cells compared to our control, which is more like 80% editing. So since there was such a low rate of editing, we sent the DNA here to MassGen for what's called deep sequencing. And we found that we were getting a rate of editing of about 0.5% or a little bit less, which is quite low for what we want. We then sorted the sequenced or the edited cells, so the bright green cells, using flow cytometry. So it's essentially a machine that separates out the different colored cells, and we individually plated each colored cell in its own well and tried to grow it. So this was about 800 individual cells that were plated. Only one cell grew, and then when we sent it to the lab, there was no editing. At this point, we also realized that we had the other unfortunate issue of ACTA2, the region around the mutation, is essentially identical to another gene called beta-actin, which is absolutely essential for cell survival. So our concern was that the base editor and the prime editor were actually editing both ACTA2 and beta-actin, which would likely be lethal to the cells that we were editing. So we stopped here, we came up with a new plan, which was introducing something called a double-stranded DNA donor. So this is also similar to CRISPR in that it makes a double-stranded DNA break on two sides. It cuts out a piece of DNA, and it perfectly drops in a double-stranded portion of DNA that we've created in the lab. And so we can introduce a new region of DNA that either has the mutation or does not have the mutation, depending on what we're trying to do. And that just sits down perfectly in that region that we've cut out. So we made two different double-stranded donors. We made one with the mutation and we made one without the mutation to correct it. And we made a guide RNA, which is what tells the double-stranded donor where to go within the genome, that has no homology, so it doesn't match that beta-actin gene. So it should do away with that risk of cutting that beta-actin, which is something that we really, really don't want to happen. We've done this for another project in our lab. So this is a double-stranded donor that created a carrier mutation. And then these are two affecteds that were created with the double-stranded donor. We have an editing efficiency in embryos right now with the double-stranded donor of about 70 to 80%. So that's quite good. And then we expect that these sheep will be born with a similar phenotype to the humans. So this was a sheep with a different genetic disease called sialidosis, which is a lysosomal storage disease that we managed last year. His name was Saltine. And he was born with quite a severe PDA. So he was managed between myself, Dr. Gray Edwards, and the boarded veterinary cardiologist at Tufts. So let me see if I can play these videos. So this is just showing.
Just click. Play it automatically.
Click through what?
Oh, just.
Oh, okay. Can that one play on the top left?
Let's click it on the presentation.
So this is just the dye being injected to show where the PDA is. And then this other video is showing the occluder device, which is the same implanter occluder that's used in children being placed. I believe this was the first time this procedure has been performed in a lamb, and he did quite well. The open approach to repairing a PDA has been done one other time in a lamb, and that lamb also did quite well. So we are very good at managing the complications that the human children undergo in farm animals. And we have a great team at Tufts between the veterinary cardiologists and the veterinary neurologists, and we feel very comfortable managing these guys after they're born. So our milestones have now changed a little bit as of yesterday. So our plan initially was to edit some embryos with the double-stranded donor and then take a look at the embryos in the lab. But we actually moved up our timeline, and yesterday we implanted 9 edited embryos in sheep. So the sheep with the mutation should be born in September. And that will be a very exciting time to have 9 ACTA2 lambs. Yeah, well, it was an unplanned thing that we decided to do this week. And so in the meantime, we're going to edit some embryos in the lab, and we'll look at them and look at the efficiency of the double-stranded donor, but we believe it'll be around the same as it's been with the other projects of 70 to 80%. So hopefully of those 9 embryos that are implanted, we'll get a handful of affected lambs born. And then this is our team. We're happy to take any questions.
Are we going to hold questions? Oh, it's okay.
Up there. Talk a bit about the variance between like the primate model versus bovine model. How much difference is there in the primate model versus this model?
I haven't looked at the bovine.
So primate models, it's just as a timeline thing. So non-human primates gestate a long time. They reach sexual maturity 3 to 5 years later. So if we had a while to do that, you could use primates. My lab works very hard not to use primates for our research. As a general rule, we only do it when the FDA tells us we have to. But as far as the vascular similarities go, if anything, the ruminant models, so sheep, pig, well, pigs aren't ruminants, they're ungulates, but those models will probably have a more severe phenotype because they have what's known as a reedy instead of having the normal cerebral vasculature. So they're going to have a large number of smaller diameter vessels. So if anything, we will expect to have a more severe cerebrovascular phenotype than a less severe cerebrovascular phenotype. Primate models are, people do them, and it's just, you just have to be really patient, which I'm not really patient, as you can see, because we just decided yesterday, kind of on a whim, or the day before yesterday, whatever it was, to just put them in instead of taking them back to the lab, and then we'll let them go and kind of gamble a little bit, and then we can do the analysis while those babies are gestating.
Sheep gestate for 4 months versus cattle are quite a lot longer.
9 months or longer, yeah. So I hope that clears up your question.
Go ahead.
One is you had a timeline up there.
Mhm.
And one is if you could just share that.
Share for the people on Zoom. Do you mind going to the microphone?
Sorry. You had a timeline?
Sure, we can.
Yeah, you had a timeline. If you
Hold
could share that and maybe take us through it and what comes next.
Sure, it has
After that-
changed a bit since yesterday.
Yeah, and that was my other question is there was a lot of
Yeah.
laughter over here as to, I guess curiosity out of
do you mind pulling that
why
slide up?
it was decided to just do this yesterday. Was there like any reason for that, for maybe for us to know?
Heather, you should say this.
technical
They work for many, many years and many things. They've been working for a year to get to here, and you can tell us why you decided.
Okay, so we've done this for other diseases. We have experience with the donors, and we had an extra number of recipient animals from another that we had more animals than we needed. And so we were faced with the option of just not using those animals and taking the embryos back to the lab, Or we could go ahead and gamble and implant them. And then the next time we can do the analysis and see how much editing we have and what it looks like on the genetic level. We can always terminate the pregnancies or just not use them. But this is kind of an accelerated way for us to try to get to the next step. As Patty alluded to this, we've had a very long year of a whole lot of things not working in the lab. And so we finally have something that seems to be working in our hands, and we're just really excited to make some progress. It's just, it really is that simple. I hope that makes sense to you. Okay,
Yeah,
good.
so it just changes the timeline. The lambs will be on the ground by September, hopefully. And then the analyzing the embryos in the lab we'll just do in the next month or so. So.
Heather or Abby, any of you, could you elaborate what is it that we do in large animals that is necessary, in particular in gene therapy? What kind of things do you get to experiment and get an estimate? If it's the dose, is it the dose that you need to use? Is it that you can or not correct, or is it to see side effects of the therapy? What do you guys use the animals? And if you can give us an example of your FDA experience of one of those factors.
Yeah, I mean, all of the above: dose, toxicity, tissue distribution within the body, which tissues we're able to correct efficiently. I'll let Heather talk about her FDA experience.
So this is a genetic therapy, right? So it has to have the same genetic sequence in order for us to predict what is going to happen. So from our perspective, that's kind of the most important thing, is to make sure that the same reagents that will be used in children and patients will be the same that we can use in the animals. So that's first and foremost why this is important, because you can test safety, but unless you have like perfect target engagement and you have that mutation and that you're trying to correct and determining whether or not the mutation is corrected, it's not really giving you a whole lot of useful information testing it in normal animals. So that's the first thing. So we've done this already for 2 human clinical trials that are ongoing now for rare genetic diseases in children. They're both pediatric neurological conditions, both using a gene therapy approach. The FDA requires the use of 2 species. They really love to see species that also, or animals that suffer from the same condition, because it, you put something perfectly healthy, in a healthy person, they may not show signs of adverse events like you would if you put something in just a little, maybe not as strong. So that's the safety perspective. From I don't know if any of you have ever heard of Tay-Sachs disease, but we have an ongoing clinical trial for Tay-Sachs disease at the moment in collaboration with some folks here at MGH. And we'll tell you that we completely changed our dosing strategy for the clinical trial from the sheep studies. So we were planning on delivering it in a certain way, and we did a sheep study and we found that we were not treating the spinal cord. And so we changed everything that we were going to do as far as delivery to make sure that we accommodated the entire central nervous system. Because we did the studies in mice, but mice aren't big enough to really appreciate how the delivery changes or what the dose needs to be. So that's the dosing perspective of it. From the FDA's perspective, in our hands, they've been amazingly helpful. I think these investigator-initiated INDs, and Patty's nodding, she's had similar experience. They kind of bend over backwards to help us make our way through to get to the clinic, and they really want to see us be successful. And they do require 2 species, so this will fit the bill. So we'll have both the rodent studies, which are phenomenal, and then we'll have the large animal studies to support it. And so between those 2 things, I think we will have a clear path, but we also want to bring something forward that we think is going to work, right? And so that's kind of really important. I work with a lot of family foundations, and fundraising and getting to a point where you're ready for a clinical trial is a very long, lengthy, painful experience. And it's, you want to make sure we're putting our best foot forward. We're not doing something that we should have given a higher dose, or we should have delivered it in a certain way. And then lastly is outcome measures. You can do the same things in sheep as you could do in people. We put them in the MRI all the time. OCT imaging, we have one of those in our animal medicine department to look at eyeballs. So we can look at the eyes of these guys. We can do all of it. Electrodiagnostics, we can do all of those things. So you really get an idea of what's going to be worth looking at, what's going to work and what's not. And then when you design your clinical trial, which is not trivial, like you were talking a little bit about objective measures and things you're going to have meaningful benefit, it gives you an idea of what to pick and what not to pick, because you're not going to be 100% successful. There are going to be things we're going to address really well, hopefully the more important parts of the things, and there might be some that we miss, and we really just want to identify where it's working and where it's not working. Cognitive testing, we do that. We have a maze. Yeah, we maze train our sheep. It's pretty entertaining. Yeah, we can do that. So there's all sorts of things that we do that you can't do in rodents, or whatever, repeat collection of tissues, and they live a long time. So you can look at something past 2 years, and see how long does it work, right? Our cow is getting close to 2 years, she's still growing. But anyway, so you can do those things. And so that's our little niche. So we're happy to be here. Yeah.
You had said that the progress parts, we're all excited about that, but if you don't mind sharing, but sorry.
You had shared the progress part, and it's very exciting to this room. But you had mentioned some of the challenges. If you don't mind just educating us on a little bit of what those challenges were, and then what got us to this progress part, like what made you make that decision, just so we sort of understand how this is going on in the animal models.
Okay, so the first approaches that we tried are brilliant, and they are state of the art, but we had trouble. They're also encoded in very large plasmids. Think of them as just too big to get in, and we had a really hard time getting those into the cells to correct the cells. So there's a two-part process. You can do what we're doing, is you can directly edit that embryo, and you can put that embryo in the sheep, which is what we just did, right? Or you can edit cells and do what's called somatic cell nuclear transfer, where you take the nucleus out of one cell that you've edited and you put it into an oocyte or an egg. And then that becomes, you have to do a bunch of epigenetic modulation, but then that becomes an embryo, and then you implant that, okay? That's harder to do. Just physically, it's just harder to do, because somatic cell nuclear transfer is inherently, a lot of the embryos don't survive. You put the nucleus in there and they don't become embryos, or you put a hole in the embryo and it dies because you put a hole in it trying to inject something inside of it. So this method is just a little bit of a faster way to get there, but it wasn't the first way that we tried. So as scientists, we're always evolving. We're always losing, and we're always evolving and trying to come up with a new way to get to the same problem. And this is basically just the third evolution. The other two ideas are brilliant and they will work eventually, but this way, it just seems to be easier for us in our hands. But the
And safer.
and potentially, well, we'll see. But yeah, essentially we're evolving.
Thank you.
I hope that cleared it up. But yeah, the difference is we're instead of just taking and very precisely editing one nucleotide, we're cutting a hole in the DNA and using another piece of DNA to basically humanize that whole region. So basically we're taking, instead of just changing one nucleotide, we're changing 30 nucleotides.
Kind of like if you lifted out a portion of train tracks and then dropped new ones in.
It's a good analogy. But it's a larger portion. Brilliant stuff, all good. It's just, you do what works. You gotta try multiple things.